Hey guys, welcome back. This is another topic review video going through an entire unit, just quick and dirty, hitting some of the main points, addressing the content objectives of those units. And so the first point for this unit that I would like to address is the one that says, analyze, explain, and complete a model showing negative and positive feedback loops and how they are used to maintain homeostasis. So I'd just like to review real quick what a positive and negative feedback loop is. And so a feedback loop in general is a stimuli, a response, and how that response impacts the stimuli. Okay? And so something happens, boo, scary ghost jumps out, right? My response is, my heart rate's, okay? So my heart's pumping really fast, right? That's the response, okay? How that impacts the stimuli determines if it's a feedback loop. Okay? And so in our body, things like body temperature are stimuli. And so if I get too hot, right, my body wants to cool down, it will sweat, and I will reduce my body temperature. Okay? That is an example of a negative feedback loop, where the response stops, inhibits, or slows down the stimuli. So most of our homeostatic mechanisms are negative feedback loops because we're trying to go back to whatever point we started at, right? So we start here, we have a stimuli, it changes where we're at, change something about us. Right? Most of the time we want to bring ourselves back to that station, right? And so whatever the stimuli is, we want to cancel it out, negative feedback loop. There are a few examples of positive feedback loops in our biology. And positive feedback loops are when the response promotes, speeds up, or further triggers the initial stimuli. And so an example of that in our biology is blood clotting. Right? If I get a cut, right, when my blood that's now out in the open is exposed to the air, chemicals in the blood start clotting the blood. The blood starts to coagulate and turn into that hard, scabby stuff, right, to try to close the wound. Those clots, in turn, trigger the same response of the blood nearby. Right? So the clotting triggers more clotting. It's a positive feedback loop. Right? So the stimuli was the clotting, the response was make more clotting. Positive feedback loop. Cool? Okay, so our next prompt that I want to look at here is develop and use a model to illustrate the key functions and structures of the nervous system, including neurons, brain, and spinal cord. Okay, so our nervous system is our body system that allows us to process the external environment and respond based on that process, right? And so we have two parts of our nervous system as advanced, evolved organisms. We have our central nervous system, which is our brain stem, and our spinal cord. And we have our peripheral nervous system, which is all the other nervous system parts in our body. So the nerves that go out to our arms, all the way down to our fingers, all the way down to my toesies, right? Going to my kidneys, my liver, my stomach, all of that stuff, all the rest of the nerves are your peripheral nervous system. Okay? Those are the two main divisions of our nervous system. Yeah. Nervous system is composed of a specific cell type called a neuron, which has a very unique structure geared towards a very fast communication style. And the neuron itself has three main parts. Okay. You have the soma, which means body, and it's just the cell body. This part looks like a regular cell. There's a nucleus, there's endoplasmic reticulum, there's mitochondria. All the stuff that all of our other cells have are inside the soma. Normal cell stuff. On the front side of the soma are these little branchy things called dendrites, and they are the receivers of the signals. And then we have this long tail-like structure called an axon. At the end of the axon, there is the synaptic terminal, and the synapse is the gap between two neurons, right? And so the way that this works is that this neuron will receive a little signal on its dendrite, boop, it will activate the neuron, the signal will get passed, boo, 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 down the axon. Okay. It hits the synaptic terminal, and then a little chemical signal is released. Okay, that chemical signal called a neurotransmitter is then passed along to the next neuron. That triggers the same response in that neuron, and the process is repeated over and over again to send signals and receive signals. Right? And so if I have my finger, right, I poke my finger, I can feel that. Okay? That is the triggering of an initial neuron in my finger. It's sensitive to touch. That passes that message all the way up my arm, up, right, into my spinal cord to reach my central nervous system. Right? Then it goes up my spinal cord and into my brain. Okay? And it's connected to different neuron processing centers in my brain. And they say, oh, that signal came from the finger, and that's a feely, touchy signal, so I know that my finger's being touched. Then the brain sends a signal back down the spinal cord, back out through the peripheral nervous system to the finger to have an appropriate response. Right? And so if this is an owie touch, the signal goes up, boom, says owie, move that finger, signal comes back down, finger moves. Very fast communication. Okay. The neuron that is feeling the initial stimuli is called a sensory neuron. It does the sensing. Right. Then we have interneurons that bring the message up to the brain. The brain processes it, sends it back down into neurons, back to the finger, where motor neurons innervate our muscles and tell our body to move. Okay? Nervous system. The next prompt I want to address is asking us to explain the role of lipids within the cell membrane. Okay? And so the lipid that matters for cell membranes are phospholipids. And phospholipids are special because they are lipid tails, these two little stick things at the bottom, that are connected to a phosphate head. The structure of this molecule is very important for the function of the cell membrane. The head, the phosphate, is polar, meaning that it is also hydrophilic. It likes water. It can deal with water. It doesn't mind water one bit. Cool? The lipids are nonpolar, making them hydrophobic. They don't like water. Right? Fats don't like water. Oils don't like water. Lipids don't like water. Okay? It also means that they don't like other polar substances just like water. The phospholipids are joined up into these bilayers, so they're kind of like the bread of a sandwich, right? So you get your bread and your bread stuck together, right? You got two breads. That's basically my cell membrane. Right? And so there's a top layer of phospholipids, the bottom layer of phospholipids, and there's a little space in between. Right? The bread is touching, but there's a little gap. And I can slide some cheese in there if I want. There's no cheese in my phospholipid bilayer though. So there's the outside and the inside of the cell. Both of those environments are aqueous, meaning they're composed mostly of water. Right? And so the outside region, the hydrophilic region, is cool with it being water. Right? Cool with it being water. But the inside, because it doesn't like water, keeps the outside environment and the inside environment separate. So the inside of the cell can stay whatever it needs to be, and it doesn't just leak stuff everywhere. And so this is very important for creating a cell. The inside of a cell has to be regulated. Otherwise, it's just the outside world. It's not any different. Okay. When we're talking about cell transport, which is things moving across this membrane, these properties also play a role. Right? So if I have a molecule that's small enough to fit between these gaps, right, and also non-polar, it can just cross this membrane, no problem. However, if the molecule is bigger than these gaps, or if the molecule is polar, it can't go through here. Okay? Which leads us to the next objective, right, which asks us to explain the difference between passive and active transport. Okay? So passive transport is achieved through diffusion. Right? Molecules will move from an area of high concentration to an area of low concentration, all on their own, due to the random nature of their movements. Right? Happens. The cell doesn't have to do anything, it just occurs. Okay? So molecules that are small and non-polar can always move passively and will always move along this concentration gradient from high to low. Larger molecules and polar molecules can also move passively if they have a protein channel to travel through. Right? So embedded in the cell membrane are basically tubes of protein. Right? And molecules can pass through these tubes. The tubes have specific shapes, so they only let certain molecules through. For instance, there's a tube that's shaped specifically for water. It's called an aquaporin. And water goes through aquaporins, even though it shouldn't be able to get across the membrane. Right? So it goes boop, 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 boop. Okay? Active transport, on the other hand, requires energy input from the cell, and typically it's used to move molecules against the concentration gradient. Right? And so if I have a low amount over here and a high amount over here, usually it would go like this, right? But if this is a molecule that the cell really wants to get inside of it, it can use energy to take this molecule and pull it in, right? Or vice versa, it could take energy to take that molecule and shove it out. So active transport is typically this. There's also endo and exocytosis, which are types of active transport, where the cell is taken in a really big molecule and it like reaches out its cell membrane and closes that molecule in the membrane and pulls it in a little bubble and it's inside. Or vice versa, it takes a really big molecule, wraps it up in its membrane and spits it out. But more often than not, active transport is this movement from a low concentration to a high concentration. Uses energy. Which segues us into this next prompt, and I think it's our last prompt for this unit. Okay? This prompt is asking us to demonstrate through a model how ATP is used to move these molecules against the concentration gradient. And so I have this beautifully constructed model here. We're looking at my blue molecules. We can see that we have a high concentration of blue molecules inside and a low concentration of blue molecules outside. But this cell really wants to suck in more blue molecules. Okay. And so embedded in the cell membrane is this protein pump right here. It's shaped in a way that's made specifically to grab blue molecules and suck them in. Okay. Its initial state, its shape from the get-go, grabs the blue molecule. Right? So it's grabbed it. It's holding on to it. In order for it to make the change in shape necessary for it to suck it in, it requires a conformational change, right? Something needs to come and change the shape of that protein. And what's going to do that is an ATP, adenosine triphosphate, okay? So it's adenosine with 3 phosphates stuck on it. The ATP comes along, touches a special region on this protein channel, and one of the phosphate groups is removed from the ATP and put onto the protein channel. Okay. Putting that phosphate group onto the protein channel changes the shape of the protein channel, and it opens it up to suck that blue molecule in. Sucks the blue molecule in. Okay. Once the blue molecule's in, that phosphate falls off, and the protein channel goes back to its original shape, ready to catch another blue molecule and suck it in when another ATP comes along. Okay. The mechanisms involved in this have to do with entropy and the fact that systems always have to be moving towards disorder, right? And so in order to power this change of shape, we need to have a favorable energy shift. And so when we talk about this in simple terms, we express it as the ATP is providing energy to move this molecule. It's not exactly the most accurate of descriptions. The shape of the ATP moving along with the shape of the protein, right, to make an ADP and this new protein channel shape is entropically favorable, right? And so it happens. And when we shift towards entropically favorable situations, we have free energy available. And so we say that that's available to do work. And so that's what's happening here. Not to make it overcomplicated, I just don't feel right just saying that it gives it energy, because it's just not accurate. Right? If you'd like to explore the notion of entropic changes and how they work within biology and the way that our systems are regulated, you can always ask me, right? I'm not going to explain it here, but you can ask me if you'd like to know about it. Or you can take AP Biology, where I talk about it more, a lot more, actually. So yeah, that's it for this review video. Review video is over. Awesome. Cool. See you guys next time. Bye.